Okay, very good afternoon, my dear friends. Welcome to today's session. So this is Dr. Padala Tirupati, Assistant Professor, Department of Botany, Government Degree College for Women, Jagityal. So today's our eminent resource person, Dr. B.RUKMINI Devi Madam Garu, Assistant Professor, Department of Botany, GDC Narsapur, Medak District. Madam has huge experience in the area of plant sciences, that is botany. Madam, please welcome and start your session. So, Madam,
thank you, sir.
Share your screen.
Thank you, sir. Thank you so much, sir.
Okay, Madam, please proceed.
Okay, sir. Good afternoon, everybody. This is B.RUKMINI Devi, Assistant Professor of Botany, working in Government Degree College, NARSAPUR. Today, I would like to explain you about life history of ALBUGO. For better understanding purpose, I want to explain this topic in two sessions. In the first session, we will discuss about vegetative morphology, structure, and asexual reproduction. And in the second session, we will discuss about the sexual reproduction of ALBUGO. So, first, I'll explain you about the ALBUGO systematic position. ALBUGO is also called as a cystopus. Coming to systematic position, ALBUGO belongs to the kingdom Mycota, and it belongs to the division Eumycota. Subdivision, it belongs to Mastigomycotina. Class, it belongs to Oomystis. Order, it belongs to Peronospores. Family, Albuginaceae. Genus, ALBUGO. In the family Albuginaceae, ALBUGO is the one and only genus. In this genus, almost 25 species are present. All these species are obligate parasites. Obligate parasites means they need one host. Inside the host only, they can complete their life cycle. So that kind of parasite is called as an obligate parasite. So almost all the ALBUGO species are obligate parasites. Mostly they will infect all the flowering plants, especially Brassicaceae members, Caparidaceae members, Amaranthaceae, Nyctazinaceae, Compositae, Convolvulaceae, Portulaceae. In these families, plants are mostly affected by this fungus ALBUGO. That's called as the white rust disease. Fungus ALBUGO causes one disease in all these flowering plants. That disease is called as a white rust disease. So here in India, this genus comprises 7 species. In this important species is ALBUGO candida. This ALBUGO candida species infects mustard plants, cabbage, radish, turnip, cauliflower, and other Brassicaceae members. In all these plants, this ALBUGO candida causes white rust disease. Another species of ALBUGO, that is ALBUGO bliti, that causes white rust disease in Amaranthaceae family members. Remaining ALBUGO species causes white rust disease in weeds as well as wild plants. All these infected plants will show some white color shiny blisters. This is the disease symptom of ALBUGO. During its sexual reproduction process, this fungus ALBUGO produces some reproductive structures. So because of the formation of these reproductive structures, this white color blisters appears during this fungal infection. Because of that appearance of white color blisters, this disease is named as white rust disease. During this infection, the infected plants will show some deformations. Overgrowth of plant parts also you will notice in this disease infection. This is about a general introduction about ALBUGO. Now we will discuss about here in this image. You can see infected plant parts. Albugo infects various parts of the plant, especially it will infect foliar leaves as well as inflorescence parts. Generally, this albugo causes some chlorotic symptoms, chlorotic lesions. Chlorotic lesions means yellow color spots will be formed. Sometimes galls also formed because of this fungal infection. Galls are nothing but tumors. Where these are produced? These are produced on the upper surface of the diseased leaf. These are the symptoms produced by the genus Albugo. Here, the genus Albugo causes deformities during this fungal infection. So, these deformities are due to hypertrophy and hyperplasia. Hypertrophy means overgrowth of any particular organ or tissue is due to increased size of that particular cell. That cell size increases because of swelling. That kind of condition is called hypertrophy condition. Sometimes these deformities are arises due to hyperplasia also. Hyperplasia means the overgrowth of one particular organ or tissue is because of increase of number of cells in that particular tissue. That condition is called as a hyperplasia condition. So these two conditions leads to deformities in this white rust disease. As we have discussed about this obligate parasite earlier, here white rust disease causing fungus is called as an obligate parasite. Obligate parasite means already we have discussed earlier. Inside living host only, this fungus completes its life cycle. That kind of parasite is called as an obligate parasite. Here during its life cycle, it produces some spores. These spores are produced during asexual reproduction as well as after completion of sexual reproduction. So, spores formation you will see in the life history of Albugo. White rust is an economically important foliar disease which is caused by Albugo species. Now we will discuss about thallus structure. What kind of morphological features shown by this Albugo thallus we will discuss now. If you observe Albugo thallus, here thallus is mostly mycelial in condition. Mycelial thallus you will see in this Albugo genus. Mycelium is branched mycelium, that too filamentous in condition. What kind of mycelium we will see? Here mycelium is a plant body, that is filamentous mycelium. What kind of mycelium you will see? Filamentous mycelium you will see. This is a branched also. It will show branching. So here thallus is mostly mycelial in condition. Here this is made up of several hyphae. This several hyphae together forms this mycelium. This mycelium is a branched mycelium which is a filamentous in nature. Here you will see cenocytic condition. Here the fungal hyphae are intercellular. That means inside the host in the intercellular regions, this fungal hyphae will grow very fastly. That is why hyphae are intercellular. Next hyphae is cenocytic in condition. Cenocytic means without formation of any septum or without formation of any cross wall, many nuclei will be formed. That condition is called cenocytic condition. Simply you can explain cenocytic condition as multinucleated aseptate condition. That condition is called as a cenocytic condition. So multinucleated condition you will see without formation of any septum or cross wall. That condition you will see in the Albugo mycelium. So here it will distributed very well in the intercellular regions of the host organism. And this mycelium will produce some outgrowths. These outgrowths are called as hastoria. Actually, the shape of outgrowths are appears like knobs. So, such kind of knob-like outgrowths are produced by this fungal mycelium. That kind of knob-like outgrowths are called as hastoria. With the help of this hastoria, this fungus absorbs nutrients from the host organism. Here, this particular fungus structure, cell structure, if you observe, actually cell wall is thick in nature. Here, this fungal filaments surrounded by thick cell wall. This cell wall is made up of cellulose. Inside the filament, if you observe, in addition to nuclei, you will find mitochondria, ribosomes, endoplasmic reticulum, Golgi complex, vacuoles, etc. All these cell organelles you will find in the cytoplasm of this albugo mycelium or filament. So all these structures you will find in the shell of albugo mycelium. Here reserve food materials are stored in the form of glycogen granules. In what form? These reserve food materials are stored in albugo in the form of glycogen granules as well as oil droplets. In the form of oil droplets as well as glycogen granules, this albugo stores its food materials. So reserve food materials of albugo is oil droplets as well as glycogen granules. Here, this fungus albugo mostly distributed in the vegetative parts of the host plant. After that, these fungal mycelia enters into the reproductive parts. Then it will reach its maturity. Then it will start its reproduction. Albugo reproduced by two methods. One method is called as asexual reproduction and another method is called as sexual reproduction. So albugo reproduced by two means. One is called asexual reproduction and another one is called sexual reproduction. Here in albugo, asexual reproduction takes place by the formation of conidia. During favorable conditions, albugo reproduced by asexual means. So whenever conditions are favorable, in such kind of favorable conditions, albugo reproduced by the formation of conidia. These conidia are produced on the conidiosporangia or zoosporangia. So, in the host organism, in the intercellular spaces, this albugo mycelium developed well. Then it reaches maturity. Then it starts its asexual reproduction. Whenever this fungus reaches maturity, then what will happen? Just below the mesophyll tissue of the leaves of host plant, just below this epidermis region, this fungal mycelium produced abundantly and it shows rapid growth. Then this fungal mycelium produces some knob-shaped or club-shaped unbranched structures. Okay, in such kind of favorable conditions, just below the epidermis of the host leaves, in the epidermis of the host leaves, just below this epidermis region, this fungal mycelium grows abundantly and it produces club-shaped structures or some unbranched knob-shaped structures it will produce. These club-shaped or unbranched knob-shaped structures are called as conidiosporangia. These are called as conidiosporangia. Here, these conidiosporangia are arranged parallel to each other. Many conidiosporangia are produced. These are arranged parallel to each other. If you observe how palisade tissue is arranged in the leaf anatomy, in such a manner, here these conidiosporangia or conidiophores will arrange parallel to each other. So like that, many conidiosporangia or conidiophores will produced by this fungal mycelium. So as a result of formation of this kind of conidiosporangia or conidiophores, this fungal mycelium produces some white color blisters or spots on the surface of the leaves. So like this, conidiosporangia or conidiophores are produced. Here, each conidiophore will produce some round shaped are globe-shaped conidiums in basipetal succession. In which fashion this conidiophores will produce this conidia? In this albugo life history, this albugo mycelium will produce as some round-shaped or globe-shaped conidia produced in basipetal succession. So these are produced exogenously. So many conidia are produced from this conidiophore. These all conidia are arranged one above the another in the form of chain. So like that, many conidia are produced by this conidiophore. All are arranged in the form of a chain. So here two adjacent conidia are attached with one sticky or glue-like structures. These sticky or glue-like structures are called as disjunctors. This kind of disjunctors are present between two adjacent conidia. So from each conidiophore, many conidia are produced. All these are arranged in the form of chain. This formation is called as basipetal formation. Basipetal succession means here older one is present towards outside and the younger one is produced at the base. That kind of formation of conidia from this conidiophore is called as basipetal succession. So each conidium comprises 5 to 8 nuclei and thick dense cytoplasm. So how many nuclei are present in each conidia? Almost 5 to 8 nuclei are present, and each conidium consists of thick dense cytoplasm. Thick dense cytoplasm. So the adjacent conidia are separated from each other by some glue-like disc-shaped structures. These are called as disjunctors. These are called as disjunctors. Whenever these conidia reaches maturity, then what will happen? Because of the formation of many conidiophores and many conidia, they will create some pressure on the leaf surface. So as a result, host plant leaves will be ruptured. So as a result, these conidiophores will be exposed to the environment. So as a result, the disjunctors, whatever it is present between two adjacent conidia, those will be disintegrated. So as a result, the conidia, whatever it is arranged in the form of chain, those are separated from each other. They will be liberated out. So as a result of this kind of liberation of this conidia from this conidiophore, they will appear on the surface of the leaf as white color blisters. So because of the appearance of this white color blisters, this disease, whatever it is caused by this albugo, is named as white rust disease. So like this, asexual reproduction takes place. So how asexual reproduction takes place in the life history of albugo? By the formation of conidiophores or conidiosporangiophores. From this conidiophores or conidiosporangiophores, some globe-shaped or round-shaped structures will be formed. Those are called as conidia. Many conidia are produced in a basipetal succession. These are attached with one another with the help of disjunctors. With the help of disjunctors. Like that, many conidia are formed from each conidiophore, and these are arranged in the form of chains. On maturity, the disjunctors will be disintegrated. So as a result of disintegration of these disjunctors, these conidia are separated from each other. They will be liberated out. So this is about formation and liberation of conidia during asexual reproduction of albugo. Once these conidia are liberated, then they have to germinate. How this germination will take place, we will see now. Here, it depends on environmental conditions and temperature. Okay, here these conidia will show two types of germination. One is called as direct germination and another one is called as indirect germination. So here two types of conidia germination you will see in albugo. In Phytophthora also you will see two types of germination. So how Phytophthora will show two types of germination? In the similar way, here Albugo also shows two types of germination. So here Conidia will germinate by two means. One is called direct germination and another one is called indirect germination. How this direct germination will take place, how this indirect germination will take place, what are the consequences of this direct and indirect germination, we will discuss now. When this direct germination will take place, whenever temperatures are high, whenever temperatures are high and conditions are dry. So during dry environmental conditions and high temperatures and moisture content is very less in the air, then temperatures are between 15 to 20 degrees. During such kind of conditions, these conidia will germinate directly. They will reach suitable host organism. Whenever susceptible suitable host organism is available, immediately these conidia will germinate directly. So as a result of this germination, direct germination, this conidium will produce one germ tube. With the help of this germ tube, this conidium enters into the host tissue through stomata or any injuries if they are present on the epidermis region of the host organism. Through those injuries or through stomata, this particular conidia enters into the host organism. Okay. After entering into the host organism, this particular conidium will produce mycelium. Okay. Like that, these will show direct germination. This is about direct germination. Another type of germination also will take place in this albugo. That germination is called as indirect germination. What is the second type of germination? That is called indirect germination. Indirect germination will take place, depends on situation. That means whenever presence of moist conditions and the temperature is very low. Okay. Low temperature means less than 15 degrees centigrade temperature. In such kind of conditions, these conidia will germinate indirectly. In this indirect germination, what will happen? The protoplasm, whatever it is present in this conidium, that will be broken down into 5 to 10 pieces. Each piece develops into one zoospore. So like that, here protoplasm divided into 5 to 10 nuclear parts. Each part further develops into one particular zoospore. So like that, here indirect germination will take place. So whenever moisture conditions are available and temperature is low, in such situations, this particular conidium absorbs water and swells in size. Then the protoplasm, whatever it is present inside this conidium, that will be divided into 5 to 10 parts. Each part metamorphosed into zoospore. Later, these zoospores will germinate. So like this, indirect germination takes place. Here, the zoospores, whatever it is produced from each conidia, these zoospores are reniform in shape. Reniform means kidney shaped. These zoospores are kidney shaped or bean shaped structures, or sometimes these may be present in the form of a pear fruit shape. Each zoospore comprises 2 flagella. Okay, these are not having any cell wall. So these are naked structures. These zoospores are naked structures, pyriform in shape or reniform in shape or bean shaped structures. These are not surrounded by any cell wall. These possess 2 flagella. So with the help of flagella, these will show movement. So this kind of motile zoospores formation will take place during indirect germination of conidia. So here this motile zoospores, whenever they will reach as a suitable host, then they will lose their flagella. Then they will produce one cell wall around itself. Then it produces one germ tube. Then it enters into the host organism through stomata. Then inside the host organism, this motile zoospore produces mycelium. Here, this mycelium also diploid in condition. So like that, indirect germination will take place in albugo. Okay. So here, already we have discussed that this motile zoospores are possess two flagella. In this two flagella, one flagellum is belongs to whiplash type, another flagellum is thin cell type of flagella. Two different types of flagellums you will see in the structure of zoospores. One is whiplash type of flagellum and another one is thin cell type of flagellum. Like that, two different types of flagellums you will see in the structure of zoospores, which are produced from the conidia during indirect germination process. During indirect germination process. Like this, asexual reproduction takes place in albugo. Next, we have to discuss about sexual reproduction. Here, how these asexual reproductive structures will form, how these will germinate, how these will reach as a suitable host organism. After reaching suitable host organism, how they will enter into the host tissue. After entering into the host tissue, how they will germinate and gives rise to new fungal mycelium. All these are explained with the help of this flowchart. So like this, albugo reproduced by asexual means. Okay. So till now what we have discussed, systematic position of albugo. Next we have discussed about introduction of albugo. Next we have discussed about vegetative morphology of albugo thallus. Then we have discussed about asexual reproduction of albugo. Now we are going to discuss about sexual reproduction. Actually, this sexual reproduction takes place during the matured stages of its life cycle. So here, this sexual reproduction takes place by oogamous method. By which method this sexual reproduction takes place? Sexual reproduction will take place by one specific method that is called as oogamous method. Okay, when this sexual reproduction takes place, when the growing season comes to an end, in such kind of situations, this albugo reproduced by sexual method. So here, whenever here growing season comes to an end, then albugo reproduced by sexual method. So here some specialized structures will be formed for this sexual reproduction. What type of sexual reproduction albugo will show? Oogamous type of sexual reproduction albugo will show. So here mostly based on the involvement of number of nuclei in this fertilization process during sexual reproduction, albugo species are classified into three types. One is called as albugo candida type, second one is called as albugo bliti type, third one is called as albugo tragopogai type. On what basis albugo species are classified into three types here? On the basis of involvement of number of nuclei in the fertilization process. On the basis of involvement of number of nuclei in the fertilization process, the albugo species classified into three types. One is albugo candida type, second one is albugo bliti type, third one is albugo tragopogai type. In first type albugo candida type, oogonium and antheridium. Oogonium means female reproductive structure, antheridium means male reproductive structure. These two structures comprises only single nucleus. Okay, single nucleus is functional in the antheridium and single nucleus is functional in the oogonium. At the final stages of its development, if both are involved in the fertilization process, means one diploid oospore will be formed. Oospore means that is nothing but zygote. That type of species are kept under one particular class that is called albugo candida. Second type albugo bliti. In this Oogonium as well as antheridium comprises many functional nuclei. So, as a result of fertilization, okay, zygote will comprises multi-nuclei. Sorry, zygote comprises many nuclei. So here zygote is a multinucleated structure. Zygote is a multinucleated structure. In albugo candida, in antheridium only one functional nucleus is there. In oogonium also only one functional nucleus is there. Both are involved in fertilization. So as a result, the zygote you will see diploid condition. Okay, that zygote is also called as a oospore. But in albugo-blatae type, oogonium as well as antheridium both consist many nuclei. These all nuclei are functional in condition. So, due to the fusion of this antheridium and oogonium, one zygote will be formed. That zygote comprises many nuclei. So, here the number of nuclei may be up to 100. So, because of the presence of multinucleated zygote, these all members are kept one special class, that is called albugo-blatae. Third one is called albugo-tragopagae. In albugo-tragopagae, oogonium as well as antheridium, at the end they will consist only one functional nucleus. But after fusion of this functional nuclei, that is one antheridium nucleus and another one is oogonial nucleus, both will undergo fertilization process. As a result, one zygote will form. But here, this zygote divides many times. So as a result, the zygote is multinucleated in condition. That kind of species all are placed in albugo-tragopagae group. Like that, 3 types you will find on the basis of involvement of a number of nuclei. Now we will discuss actual sexual reproduction process in albugo. Here, in albugo, male reproductive structure and female reproductive structure are developed from the vegetative mycelium. These two are produced side by side. Okay, vegetative mycelium, apical regions produces this antheridium as well as oogonium. These are produced in the intercellular spaces of the host organism. These two sex organs are lysed side by side. So these two sex organs comprises high amount of cytoplasm as well as many nuclei. Like that, male reproductive structure antheridium and female reproductive structure oogonium will produced in albugo life history. Okay, coming to antheridium. Antheridium is a elongated club-shaped structure. What kind of structure it is? Elongated club-shaped structure. It is multinucleated in condition. That means number of nuclei are 6 to 12. But out of this, only one is remains functional at the time of fertilization. Remaining may be degenerated. In the beginning, it is multinucleated in condition. That means this particular antheridium comprises 6 to 12 nuclei. But at the time of maturation, it comprises only single nucleus. That is the functional nucleus. This functional single nucleus involves in the fertilization. So here, what kind of fertilization will take place here? Paragenous type of fertilization. So here, this antheridium oogonium are lysed side by side. So these two are come into contact with each other easily during fertilization process. Now we will discuss about oogonium structure. Oogonium structure is also here. From a vegetative mycelium, one globular structure will develop. This is multinucleated structure. That kind of a globular multinucleated structure which is produced from the vegetative mycelium of the albugo is called as oogonium. This comprises thick cytoplasm. Okay, in this thick dense cytoplasm, almost 100 nuclei are present. Like that, oogonium will develop. In this oogonium, The protoplasm is differentiated into two important parts. One is called central ooplasm and another one is called peripheral periplasm. So the protoplasmic content, whatever it is present in the oogonium, that will be differentiated into two important regions. One is called central ooplasm region and another one is peripheral periplasm region. So central ooplasm region comprises thick dense cytoplasm that comprises one definite conspicuous nucleus. Okay, this will functions as oosphere, are also called as egg cell. Okay, this is functional conspicuous single nucleus, whatever it is present in the ooplasm region of the oogonium, that is called as oosphere. Okay, this periplasm comprises many small nuclei. In addition to this many small nuclei, cytoplasm also consists many vacuoles. So like this periplasm region is present. Okay, like this oogonium will form and antheridium also produced. Later, next step is fertilization. Okay, how this fertilization will take place? What kind of fertilization will take place? After fertilization, what kind of events will take place? All these we will discuss in the next session. Sir?
Madam, only 3 minutes left. Only 2.5 minutes left, madam.
Okay, sir. అది ఇంకా కొంచెం టైం పడుతుంది సార్, నెక్స్ట్ క్లాస్ లో కంటిన్యూ చేద్దాం.
నెక్స్ట్ క్లాస్ కంటిన్యూ చేద్దాం. ఇప్పుడు ఆపేద్దామా మేడం?
ఓకే సార్, ఆపేద్దాం సార్.
రైట్ మేడం, థ్యాంక్ యూ. ఓకే, థ్యాంక్ యూ వెరీ మచ్.
థ్యాంక్ యూ సార్.
Nice explanation, wonderful presentation. Thank you, madam. Tomorrow we will meet.
Thank you, sir.
Right.
Okay, sir.